Welcome to Sleepy Science Odyssey. Tonight, plants. The multicellular organisms that transformed Earth's atmosphere and landscape, from the first mosses colonizing bare rock to the towering trees dominating forests, from desert cacti surviving months without water to aquatic plants thriving underwater, from carnivorous species trapping insects to parasitic plants stealing nutrients from hosts. We'll explore how plants capture sunlight and convert it to chemical energy, how they've adapted to nearly every environment on Earth, how they communicate and defend themselves, and why understanding plant biology matters for understanding life itself. If you're new here, this channel is for people who need something to think about before they can stop thinking. Real plant science, no exaggeration, just quiet facts in the dark. Let's begin. Multicellular photoautotrophs evolved from freshwater green algae approximately 450 to 500 million years ago. The colonization of land by plants represents one of the most important events in Earth's history, transforming barren rocky landscapes into the green world we know today. Before plants, terrestrial environments were largely lifeless, except for microbial films on rocks and in soils. The evolution of land plants from aquatic ancestors required numerous innovations allowing survival outside water, a challenge comparable to animals' later transition from water to land. The ancestors of land plants were freshwater green algae belonging to the charophyte group, which still exist today. These algae possessed features pre-adapting them for terrestrial life, including cell walls containing cellulose, chloroplasts with similar photosynthetic pigments to land plants, and certain reproductive features. The transition to land required additional innovations including cuticles, waxy coatings reducing water loss from surfaces, stomata, adjustable pores allowing gas exchange while minimizing water loss, and anchoring structures replacing the buoyancy that water provided. The earliest land plants were small, moss-like organisms lacking vascular tissues to transport water and nutrients, limiting them to moist environments and small sizes. These bryophytes, mosses, liverworts, and hornworts still exist today, comprising approximately 20,000 species that occupy moist habitats globally. Bryophytes lack true roots, instead using rhizoids, simple hair-like structures, for anchoring to substrates. They lack leaves with vascular tissue, instead having simple leaf-like structures that are typically only one cell thick, requiring that all cells have direct access to water from the environment. The evolution of vascular tissue, xylem conducting water and dissolved minerals from roots upward, and phloem conducting sugars and other organic compounds throughout the plant, revolutionized plant biology by allowing plants to grow tall and occupy diverse habitats. The first vascular plants appeared approximately 420 million years ago, initiating rapid diversification and increasing dominance of terrestrial landscapes. These early vascular plants were relatively simple, lacking leaves and seeds, but their descendants would evolve the enormous diversity characterizing modern plant life. Lycophytes, club mosses, and relatives represent an ancient lineage of vascular plants that were dominant during the Carboniferous period 300 to 360 million years ago, when some species grew as trees reaching heights of 40 meters and forming extensive forests. Today's lycophytes are much smaller, mostly herbaceous plants growing in tropical rainforests and temperate woodlands, comprising approximately 1,200 species. Despite their common name, club mosses, they are not true mosses, but vascular plants more closely related to ferns and seed plants. Monilophytes, ferns and horsetails, represent another major group of seedless vascular plants, comprising approximately 12,000 species that occupy diverse habitats from tropical rainforests to Arctic tundra, though most diverse in wet tropical environments. Ferns are characterized by large, often compound leaves called fronds that typically uncurl from coiled structures called fiddleheads during development. Like bryophytes and lycophytes, ferns reproduce via spores rather than seeds and require water for fertilization, with swimming sperm that must reach eggs in moist conditions. The evolution of seeds approximately 360 million years ago freed plants from dependence on water for reproduction, allowing colonization of drier habitats and contributing to the diversification of terrestrial ecosystems. Seeds package embryos with stored nutrients and protective coats, allowing dispersal and dormancy until conditions favor germination. The earliest seed plants were gymnosperms, naked seed plants including conifers, cycads, ginkgo, and netophytes, which produce seeds not enclosed in fruits. Gymnosperms dominated terrestrial vegetation from roughly 300 to 140 million years ago and remain important components of many ecosystems, particularly temperate and boreal forests dominated by conifers. Conifers, pines, spruces, firs, and relatives comprise approximately 630 species and include the tallest trees, the most massive trees, and some of the oldest living organisms. Their success reflects adaptations to cold and dry conditions, including needle-like leaves with low surface area reducing water loss, and ability to photosynthesize at low temperatures when deciduous trees are dormant. Flowering plants, or angiosperms, appeared approximately 140 million years ago and diversified rapidly, becoming dominant in most terrestrial habitats by 100 million years ago, a pattern Darwin called an abominable mystery because the fossil record showed such sudden diversification. Today, angiosperms comprise approximately 350,000 to 400,000 species, over 90% of all plant species, occupying nearly every terrestrial habitat and providing most human food, fiber, medicine, and timber. The key innovations of angiosperms include flowers, structures that facilitate pollination through diverse mechanisms including wind, water, and especially animals, and fruits, structures that protect seeds and facilitate dispersal. These features, combined with more efficient vascular systems, rapid reproduction, and diverse growth forms, have made angiosperms extraordinarily successful. Angiosperms are divided into several major groups with monocots, grasses, orchids, palms, and relatives with approximately 70,000 species, and eudicots, roses, oaks, sunflowers, and relatives with approximately 200,000 species, being the largest. The fossil record reveals that early land plants faced extreme challenges that shaped their evolution. The Devonian period, approximately 420 to 360 million years ago, saw the emergence of the first forests, dominated by tree-sized lycophytes and early seed plants. These forests fundamentally altered Earth's climate by accelerating weathering of rocks through root activity and organic acids, which drew down atmospheric carbon dioxide levels. This carbon dioxide reduction, combined with the burial of vast quantities of organic matter in swamps, material that would eventually become coal deposits, contributed to global cooling and possibly glaciation events. The Carboniferous period, 360 to 300 million years ago, takes its name from the extensive coal deposits formed from plant material during this time. The forests of this period were dominated by giant lycophytes, including lepidodendron, scale trees reaching 30 to 40 meters tall, and sigillaria, along with tree ferns and early seed plants. These forests thrived in vast swamps covering much of the equatorial regions, creating conditions where plant material accumulated faster than it could decompose. The low oxygen levels in these swamps prevented complete decay, allowing organic matter to accumulate and eventually transform into coal through heat and pressure over millions of years. The atmospheric oxygen concentration during the Carboniferous reached levels as high as 35%, significantly higher than today's 21%, largely due to the extensive plant growth and organic matter burial that removed carbon dioxide while adding oxygen. This elevated oxygen supported giant arthropods, including dragonflies with wingspans exceeding 70 centimeters and millipedes, reaching 2 meters in length. The high oxygen also increased fire risk, with evidence from fossil charcoal suggesting that wildfires were common despite the swampy conditions, creating a dynamic ecosystem shaped by recurring fires. The evolution of lignin, the complex phenolic polymer that provides structural support in vascular plants, was crucial for the development of tall plants and woody stems. Lignin strengthens cell walls, allowing plants to grow upright against gravity and resist wind stress. However, during the Carboniferous, organisms capable of efficiently decomposing lignin had not yet evolved or were not abundant, contributing to the accumulation of plant material in swamps. The evolution of lignin-degrading fungi, particularly white rot fungi capable of breaking down lignin using specialized enzymes, took tens of millions of years, during which vast quantities of lignin-rich plant material accumulated as coal. Photosynthesis converts light energy plus carbon dioxide and water into glucose and oxygen. Photosynthesis, the process by which plants convert solar energy into chemical energy stored in organic molecules, is arguably the most important biological process on Earth. It produces essentially all the organic matter forming the base of food webs and releases the oxygen that most organisms require for respiration. Understanding photosynthesis is fundamental to understanding how life on Earth functions and how human activities, including agriculture and climate change, affect the biosphere. The overall equation for photosynthesis is deceptively simple. 6 carbon dioxide plus 6 water plus light energy yields 1 glucose plus 6 oxygen. However, this summary conceals enormously complex biochemistry involving hundreds of enzymes, multiple membrane systems, and intricate regulatory mechanisms. Photosynthesis occurs in 2 stages: the light-dependent reactions occurring in thylakoid membranes and the light-independent reactions, or Calvin's cycle, occurring in the stroma, the fluid-filled space surrounding thylakoids. Chloroplasts, the organelles where photosynthesis occurs, are descendants of ancient cyanobacteria that were engulfed by eukaryotic cells approximately 1 to 1.5 billion years ago in an endosymbiotic event. This origin explains why chloroplasts retain their own DNA, ribosomes, and double membranes: the inner membrane from the original bacterium and outer membrane from the host's engulfing vesicle. A typical plant cell might contain 20 to 100 chloroplasts, with numbers varying by cell type and environmental conditions. The light-dependent reactions begin when photons strike photosynthetic pigments in thylakoid membranes. The primary pigment, Chlorophyll absorbs strongly in blue and red wavelengths while reflecting green, giving plants their characteristic color. Accessory pigments, including chlorophyll B and carotenoids, absorb additional wavelengths, extending the range of light usable for photosynthesis and providing photoprotection by dissipating excess light energy that could damage the photosynthetic machinery. When chlorophyll absorbs light, electrons become excited to higher energy states. These energized electrons pass through an electron transport chain in thylakoid membranes, driving the synthesis of ATP, the universal energy currency of cells, and NADPH, a reducing agent providing electrons for biosynthesis. This process, called photophosphorylation, is analogous to oxidative phosphorylation in mitochondria, but runs in reverse, using light energy to drive unfavorable electron transfers rather than extracting energy from favorable transfers. A crucial feature of the light-dependent reactions is photolysis, the splitting of water molecules to replace electrons lost from chlorophyll. This process releases oxygen as a byproduct, explaining why photosynthesis produces the oxygen we breathe. Over geological time, photosynthesis transformed Earth's atmosphere from oxygen-poor to oxygen-rich, enabling the evolution of aerobic respiration and complex multicellular life. The current atmospheric oxygen concentration of approximately 21% represents a balance between photosynthetic production and respiratory consumption. The light-independent reactions, or Calvin's cycle, use the ATP and NADPH produced by light-dependent reactions to fix carbon dioxide into organic molecules. The key enzyme is ribulose 1,5-bisphosphate carboxylase oxygenase, abbreviated RuBisCO, which catalyzes the attachment of carbon dioxide to a 5-carbon sugar called ribulose 1,5-bisphosphate. The resulting 6-carbon compound immediately splits into 2 3-carbon molecules that, through a series of reactions, are converted to glyceraldehyde 3-phosphate, which serves as the starting material for synthesizing glucose and other organic compounds. RuBisCO is remarkable for several reasons. It is the most abundant protein on Earth, comprising up to 50% of leaf protein content, which makes sense given that it catalyzes the entry of inorganic carbon into the biosphere. However, it is also one of the slowest enzymes known, fixing only about 3 to 10 carbon dioxide molecules per second, millions of times slower than typical enzymes. This explains why plants produce such enormous quantities of it. Additionally, RuBisCO has imperfect specificity, sometimes fixing oxygen instead of carbon dioxide in a process called photorespiration that wastes energy and releases previously fixed carbon dioxide. Photorespiration increases with temperature and decreases with carbon dioxide concentration, becoming particularly problematic in hot, dry conditions when stomata close to conserve water, reducing internal carbon dioxide and increasing the oxygen to carbon dioxide ratio. Some estimates suggest photorespiration can reduce photosynthetic efficiency by 20 to 50% under typical conditions, representing a significant cost. This inefficiency has driven the evolution of alternative photosynthetic pathways that minimize photorespiration. C4 photosynthesis evolved independently at least 60 times in approximately 46 plant families, particularly in tropical grasses including corn, sugarcane, and sorghum. In C4 plants, carbon dioxide is initially fixed by a different enzyme, phosphoenolpyruvate carboxylase, which has much higher affinity for carbon dioxide than RuBisCO and no oxygenase activity. This initial fixation produces 4-carbon compounds, hence the name C4, which are transported to specialized bundle sheath cells where carbon dioxide is released in high concentrations around RuBisCO, saturating it with carbon dioxide and suppressing photorespiration. This spatial separation of initial carbon fixation and the Calvin cycle requires distinctive leaf anatomy with 2 photosynthetic cell types arranged concentrically around vascular bundles, mesophyll cells surrounding bundle sheath cells. This Krantz anatomy is visible under microscopes and allows identification of C4 plants from leaf structure. The C4 pathway requires additional energy compared to C3 photosynthesis, 2 ATP per carbon dioxide fixed, but this cost is offset by eliminating photorespiration losses in hot, bright conditions. C4 plants dominate tropical grasslands and are important agricultural crops in warm climates. CAM photosynthesis, crassulacean acid metabolism, named for the Crassulaceae family where it was first discovered, represents another solution to photorespiration, particularly important in arid environments. CAM plants open stomata at night when temperatures are cooler and humidity higher, taking up carbon dioxide and fixing it into 4-carbon organic acids that are stored in vacuoles. During the day, stomata close to conserve water, and the stored acids release carbon dioxide internally in high concentrations around RuBisCO, again suppressing photorespiration. This temporal separation of carbon dioxide uptake and the Calvin cycle contrasts with C4's spatial separation, but achieves similar results, concentrating carbon dioxide around RuBisCO. CAM plants include cacti, agaves, pineapples, and many epiphytes, reflecting adaptations to water-limited conditions. The extreme water use efficiency of CAM photosynthesis, up to 6 times more efficient than C3 plants, allows CAM species to inhabit deserts and other dry habitats where C3 plants cannot survive. However, CAM plants typically have low growth rates because carbon dioxide uptake is limited to nighttime hours when stomata are open. Photosynthesis rates vary enormously with environmental conditions, exhibiting responses to light, temperature, water availability, and nutrients. The relationship between light intensity and photosynthesis rate shows characteristic shape. At low light, photosynthesis increases linearly with light intensity, but at high light, photosynthesis saturates as other factors become limiting. The light intensity where photosynthesis equals respiration is called the compensation point, typically 1 to 5% of full sunlight for shade-tolerant species, and 5 to 20% for sun-adapted species. The light intensity where photosynthesis saturates is called the saturation point, varying from 20 to 30% of full sunlight for shade-tolerant species to requiring full sunlight for sun-adapted species. Temperature affects photosynthesis through its effects on enzyme activities and water relations. Each species has an optimal temperature range where photosynthesis is maximal, typically 20 to 30 degrees Celsius for C3 plants from temperate climates, 30 to 40 degrees for C4 tropical plants, and 15 to 25 degrees for CAM plants. Above optimal temperatures, photosynthesis declines due to enzyme denaturation, increased photorespiration in C3 plants, and water stress from high evaporative demands. Below optimal temperatures, photosynthesis declines due to reduced enzyme activities, and at freezing temperatures, ice crystal formation damaging cell structures. Water stress reduces photosynthesis both directly through effects on photosynthetic biochemistry and indirectly through stomatal closure, reducing carbon dioxide uptake. As soil water declines, plants close stomata to prevent dehydration, but this reduces internal carbon dioxide concentrations, limiting photosynthesis. Different species show different sensitivities to water stress, with some closing stomata at modest water deficits to conserve water, a conservative water use strategy, while others maintain open stomata longer, maximizing photosynthesis at risk of severe dehydration, a profligate water use strategy. Nutrient availability, particularly nitrogen and phosphorus, strongly affects photosynthesis because these nutrients are required for synthesizing chlorophyll, RuBisCO, and other photosynthetic proteins. Nitrogen-deficient plants show reduced chlorophyll content, appearing pale yellow-green rather than deep green, and have reduced photosynthetic capacity. Phosphorus deficiency reduces ATP synthesis, limiting light-dependent reactions. Other nutrients, including magnesium, iron, and manganese, are also essential for photosynthesis, with deficiencies causing specific symptoms including chlorosis, yellowing, and necrosis, tissue death. The quantum yield of photosynthesis, the number of carbon dioxide molecules fixed per photon absorbed, provides a measure of photosynthetic efficiency. Under optimal conditions with low light intensity, quantum yields can approach the theoretical maximum of approximately 0.1, meaning one carbon dioxide molecule is fixed for every 10 photons absorbed. This efficiency reflects the fundamental biochemistry of photosynthesis, where multiple photons are required to drive the electron transport chains, generating ATP and NADPH. At high light intensities, quantum yields decline as other factors become limiting, and excess light energy must be dissipated as heat to prevent damage. Photoinhibition occurs when light intensity exceeds the capacity of photosynthetic machinery to process energy, causing damage to photosystem proteins, particularly the D1 protein in photosystem 2. This damage occurs continuously even under normal light conditions, but plants have repair mechanisms that replace damaged proteins faster than damage accumulates. Under excessive light, damage outpaces repair, causing net photoinhibition that reduces photosynthetic capacity. Shade-adapted plants are particularly susceptible to photoinhibition because their repair mechanisms are tuned to low light conditions and cannot keep pace with damage under high light. Photoprotective mechanisms help plants cope with excess light energy. The xanthophyll cycle involves reversible conversion of violet xanthan to zeaxanthin under high light, with zeaxanthin dissipating excess energy as heat through non-photochemical quenching. This thermal dissipation prevents formation of reactive oxygen species that would damage cellular components. Plants also adjust chloroplast positions within cells, moving them to cell edges to reduce light absorption when light is excessive, and moving them to maximize light interception when light is limiting. Leaf movements, including leaf rolling or folding, also reduce light interception under stress conditions. Root systems anchor plants, absorb water and nutrients, and interact with soil organisms. Roots, the typically underground organs that anchor plants and absorb water and nutrients, are as important for plant function as the more visible shoots, yet they receive less attention because they're hidden beneath soil. Root systems can be enormous, sometimes exceeding shoot systems in mass, and certainly exceeding them in surface area, when fine roots and root hairs are considered. Understanding root biology is crucial for understanding plant ecology, agriculture, and responses to environmental change. Root architecture, the spatial configuration of root systems, varies among species, reflecting different evolutionary strategies and adaptations to soil conditions. Taproot systems feature a dominant central root growing downward with smaller lateral roots branching from it. Many dicots, including carrots, dandelions, and oak trees, develop taproots, which can extend many meters deep, accessing water and nutrients unavailable to shallow roots. Some desert plants develop taproots exceeding 20 meters depth, with records of Boscia albitrunca roots reaching over 60 meters in the Kalahari Desert, allowing access to deep groundwater. Fibrous root systems consist of many similarly sized roots spreading from the stem base without a dominant central root. Grasses and most monocots develop fibrous systems, which typically occupy shallower soil than taproots, but spread laterally over large areas. A mature corn plant might have fibrous roots extending 1 to 2 meters depth and spreading 1 to 2 meters laterally, while prairie grasses can develop roots reaching 3 to 4 meters depth despite shallow initial growth. The extensive fibrous roots of grasses make them effective at soil stabilization, preventing erosion on slopes and disturbed sites. Adventitious roots arise from stems or leaves rather than from the primary root or its branches. Many plants produce adventitious roots under specific conditions, including flooding, burial, or wounding. Some species, including ivy, mangroves, and many tropical rainforest trees, routinely produce adventitious roots that serve specialized functions. Aerial roots growing from above-ground stems are adventitious roots that may serve mechanical support, as in corn prop roots and mangrove stilt roots, or absorption of water and nutrients, as in epiphytic orchids and bromeliads. Root growth occurs through cell division in apical meristems located near root tips, protected by root caps, thimble-shaped structures covering meristems. Root caps contain statoliths, dense starch-filled organelles that settle to the bottom of cells under gravity, providing information about gravitational direction that guides positive gravitropic growth, roots growing downward. Behind meristems, cells elongate rapidly, pushing root tips through soil. Behind the elongation zone, cells differentiate into specialized tissues including epidermis, cortex, endodermis, and vascular tissues. Root hairs, tubular extensions of epidermal cells, dramatically increase root surface area for water and nutrient absorption. A single rye plant was estimated to have approximately 14 billion root hairs, with total surface area exceeding 600 square meters, more than 100 times the leaf surface area. Root hairs are ephemeral, living only days to weeks, but are continuously replaced as roots grow, maintaining absorptive surface area. The density of root hairs increases in response to nutrient deficiency, representing adaptive plasticity that improves foraging. Water uptake by roots occurs passively down water potential gradients from soil to root to shoot to atmosphere, driven ultimately by evaporation from leaves. Water can follow multiple pathways through roots. The apoplastic pathway through cell walls without crossing membranes, the symplastic pathway through cytoplasm connected by plasmodesmata, channels linking adjacent cells, and the transmembrane pathway crossing membranes repeatedly. All pathways eventually pass through the endodermis, where Casparian strips, waterproof bands in cell walls, force water into the symplastic pathway, allowing selective uptake and preventing backflow. Nutrient uptake requires active transport because nutrient concentrations are typically higher in roots than in soil, requiring energy to move nutrients against concentration gradients. Different nutrients are transported by different membrane proteins with varying specificities and affinities. Nitrogen is taken up as either ammonium ions or nitrate ions, with different transporters for each form. Phosphorus is taken up as phosphate ions through high-affinity transporters that are induced under phosphorus deficiency. Potassium, calcium, magnesium, sulfur, and micronutrients each have specific transport systems, with regulation adjusting uptake rates to match plant needs. The soil surrounding roots quickly becomes depleted of mobile nutrients like nitrate, creating depletion zones where nutrient concentrations are lower than bulk soil. The extent of depletion zones depends on nutrient mobility and root uptake rates, with depletion zones for nitrate extending several millimeters from roots, while depletion zones for immobile nutrients like phosphate extend only fractions of a millimeter. This creates strong selection for root traits that maximize soil exploration, including rapid elongation, profuse branching, and association with mycorrhizal fungi that extend beyond depletion zones. Mycorrhizae, symbiotic associations between plant roots and fungi, occur in approximately 90% of plant species, significantly increasing nutrient uptake, particularly phosphorus. Plants allocate 10 to 30% of photosynthetic production to mycorrhizal fungi, representing substantial carbon cost, but receive benefits exceeding costs through improved nutrition and other services. Arbuscular mycorrhizae, formed by glomeromycete fungi, penetrate root cells and form branching structures called arbuscules where nutrient exchange occurs. These are the most common mycorrhizal type, found in approximately 80% of plant species across most plant families. Ectomycorrhizae, formed by basidiomycete and ascomycete fungi, surround root tips with fungal sheaths and penetrate between root cells without entering them. Ectomycorrhizae are found primarily in temperate and boreal forest trees, including pines, oaks, birches, and beeches, involving approximately 20,000 to 25,000 fungal species. The fungal mantles covering root tips can be recognized visually, often giving roots distinctive colors and branching patterns. Some ectomycorrhizal fungi produce large fruiting bodies, mushrooms, above ground, representing only small fractions of the extensive hyphal networks below ground. Mycorrhizal fungi extend far beyond roots, exploring large soil volumes inaccessible to roots alone. A single plant may be connected to fungal networks containing hundreds of meters of hyphae per gram of soil. These networks not only increase nutrient uptake, but also connect multiple plants, potentially allowing resource transfer between individuals. Evidence shows that carbon, nitrogen, and phosphorus can flow through mycorrhizal networks between plants, with implications for competition, facilitation, and community dynamics. Some research suggests parent trees may support offspring through mycorrhizal connections, though the extent and significance of such transfers remains debated. Root architecture shows remarkable plasticity in response to nutrient availability, with preferential growth toward nutrient-rich patches. When roots encounter nutrient-rich zones, they proliferate through increased branching and elongation, creating localized dense root systems that exploit patches. This foraging behavior allows plants to capture heterogeneously distributed resources more effectively than uniform root growth would permit. The mechanisms involve local sensing of nutrient concentrations by roots, triggering hormonal signals that modify growth patterns. Competition among roots of neighboring plants is intense but invisible, occurring beneath soil where observation is difficult. Plants compete for water and nutrients through both symmetric competition, where larger plants capture proportionally more resources, and asymmetric competition, where larger plants capture disproportionately more resources by excluding smaller plants from access. Root competition can be as or more important than shoot competition in determining plant performance and community composition, though it receives less research attention because of methodological challenges. Roots also produce and release numerous organic compounds into soil through exudation, a process that represents significant carbon cost but provides various benefits. Root exudates include sugars, amino acids, organic acids, enzymes, and secondary metabolites that modify the rhizosphere, the narrow zone of soil surrounding roots. These exudates attract beneficial microbes, suppress pathogens, mobilize nutrients from soil minerals, and potentially communicate with neighboring plants. The composition of exudates varies among species and with nutrient status, representing a mechanism through which plants engineer their local soil environments. Deep rooting allows access to water and nutrients unavailable to shallow roots, providing advantages during droughts when surface soils dry. However, deep rooting requires substantial carbon investment in long roots that traverse nutrient-poor subsoils, creating a trade-off between drought tolerance and growth rate. Some perennial species invest heavily in deep roots, providing insurance against droughts at cost of slow growth, while many annual species maintain shallow roots, gambling on adequate surface moisture. Climate change is altering these trade-offs, potentially favoring deep-rooted species in regions experiencing increased drought frequency. Fine root turnover, the continuous death and replacement of fine roots, represents major but often overlooked carbon flux from plants to soils. Fine roots typically live only weeks to months before dying and being replaced, with turnover rates sometimes exceeding 100% annually. This rapid turnover means that annual fine root production can equal or exceed above-ground production, representing 20 to 50% of total plant photosynthetic production. Dead roots provide substantial organic matter inputs to soil, fueling decomposer communities and contributing to soil carbon storage. The total length of roots in a single plant can be staggering. A mature rye plant grown in controlled conditions was found to have a total root length exceeding 620 kilometers, approximately the distance from New York to Detroit, with an additional 10,000 kilometers of root hairs. This enormous total length, packed into a relatively small soil volume, illustrates the intensive exploration of soil by plant roots. While such extreme values come from optimal growing conditions and likely exceed field-grown plants, they demonstrate the potential scale of root systems. Hydraulic redistribution, the movement of water from moist to dry soil through plant roots, represents an important but often overlooked phenomenon. Plants with roots spanning soil layers with different moisture contents can absorb water from moist deep soil at night when transpiration is low, then release some of this water into dry surface soil through their roots. This nocturnal hydraulic redistribution can maintain fine roots in dry surface soil, facilitate nutrient uptake, and benefit neighboring plants sharing the same soil volume. The extent of hydraulic redistribution varies among species and environmental conditions, with greatest effects in ecosystems experiencing seasonal drought. Root nodules, specialized structures housing nitrogen-fixing bacteria, occur in legumes and several other plant families, including alders and actinorhizal plants. The bacteria, primarily rhizobium species in legumes and frankia in actinorhizal plants, convert atmospheric nitrogen gas into ammonia that plants can use, providing nitrogen nutrition in nitrogen-poor soils. In exchange, plants provide carbohydrates to bacteria, creating mutualistic symbioses. Nodule formation involves complex signaling between plants and bacteria, with plants releasing flavonoid compounds that attract bacteria and bacteria releasing nod factors that trigger nodule development in roots. The energetic cost of biological nitrogen fixation is substantial. Fixing 1 kilogram of nitrogen requires approximately 20 to 30 kilograms of carbohydrate, representing significant carbon investment. However, this cost is often less than the cost of developing extensive root systems to capture soil nitrogen or the competitive disadvantage of nitrogen limitation. Legumes typically fix 50 to 300 kilograms of nitrogen per hectare per year, with fixation rates depending on species, environmental conditions, and soil nitrogen availability. When soil nitrogen is abundant, many legumes reduce or cease fixation, reflecting the energetic cost. Leaves are specialized organs optimized for light capture and gas exchange. Leaves, the primary photosynthetic organs of most plants, exhibit extraordinary diversity in size, shape, structure, and function, reflecting adaptations to vastly different light, temperature, and moisture conditions across environments. Despite this diversity, all leaves face the fundamental challenge of maximizing carbon dioxide uptake for photosynthesis while minimizing water loss through transpiration, a trade-off that has shaped leaf evolution and continues to constrain plant performance. Leaf anatomy reflects the dual requirements of light capture and gas exchange. The upper epidermis is typically covered with a waxy cuticle that reduces water loss, with few or no stomata. Beneath the epidermis lies the palisade mesophyll, columnar cells packed with chloroplasts arranged to maximize light interception, where most photosynthesis occurs. Below the palisade layer lies spongy mesophyll, irregularly shaped cells with large air spaces facilitating gas diffusion between stomata and photosynthetic cells. The lower epidermis contains most stomata, typically 50 to 500 per square millimeter, depending on species, and is also covered with cuticle. Stomata are remarkable structures consisting of 2 guard cells surrounding a pore that can open and close, regulating the trade-off between carbon dioxide uptake and water loss. Guard cells are shaped like paired sausages, with thicker walls on the inner faces bordering the pore. When guard cells absorb water and become turgid, the uneven wall thickening causes them to bow outward, opening the pore. When guard cells lose water and become flaccid, they straighten, closing the pore. This simple mechanical system allows precise control of gas exchange in response to environmental conditions and plant water status. Stomatal opening and closing is controlled by multiple signals including light, carbon dioxide concentration, humidity, and the hormone abscisic acid. Light promotes opening through photosynthetic electron transport in guard cell chloroplasts, which activates proton pumps that accumulate potassium ions in guard cells, drawing in water osmotically. High carbon dioxide promotes closing, providing negative feedback that prevents excessive carbon dioxide uptake. Low humidity promotes closing, reducing water loss when evaporative demand is high. Abscisic acid, produced in roots experiencing water stress, signals stomata to close, conserving water when soil moisture is limiting. The density and distribution of stomata vary enormously among species, and with growing conditions, reflecting adaptations to different environments. Most species have more stomata on lower leaf surfaces, hypostomata, which reduces heating from direct sunlight and may reduce water loss. Some species have stomata only on lower surfaces, while others have similar densities on both surfaces, amphistoma, which may allow higher gas exchange rates. Aquatic plants often have stomata only on upper surfaces where they contact air. Stomatal density typically increases in low carbon dioxide and decreases in high carbon dioxide, both evolutionary responses to changing atmospheric composition and plastic responses during individual development. Leaf size varies over 6 orders of magnitude, from less than 1 square millimeter in some alpine plants to over 10 square meters in some palms and aroids, making leaf size among the most variable of plant traits. This variation reflects trade-offs among multiple functions. Large leaves capture more light per unit construction cost, but heat up more in sunlight due to thicker boundary layers, the layers of still air adjacent to surfaces that reduce convective cooling. Small leaves maintain temperatures closer to air temperature, preventing overheating in high light and hot conditions, but have higher construction costs per unit area due to greater leaf margin length per unit area. The worldwide pattern of leaf size reflects these biophysical constraints. Tropical rainforest plants have large leaves because high humidity and relatively low light favor large size. Temperate forest plants have intermediate-sized leaves reflecting moderate conditions. Desert plants have small leaves to prevent overheating, and Arctic alpine plants have small leaves to warm above air temperature for photosynthesis. This predictable relationship between leaf size and climate allows reconstruction of past climates from fossil leaf assemblages, with larger mean leaf sizes indicating warmer, wetter climates. Leaf longevity varies from weeks in deciduous herbs to over 40 years in some evergreen conifers and broad-leaved trees, representing a fundamental axis of variation in plant strategies. Short-lived leaves typically have high photosynthetic rates per unit mass, rapid nutrient cycling, and low construction costs, allowing rapid growth in favorable conditions. Long-lived leaves typically have lower photosynthetic rates, but recoup their costs over multiple growing seasons, have higher construction costs, including physical and chemical defenses against herbivores and pathogens, and retain nutrients longer, reducing nutrient requirements. These contrasting strategies create a leaf economic spectrum describing correlated variation in leaf traits across species. Deciduous leaves are shed seasonally, typically in autumn in temperate regions or at the onset of dry seasons in tropical regions. Before abscission, nutrients including nitrogen, phosphorus, and potassium are resorbed from senescing leaves and transported to storage organs, allowing plants to recycle 50 to 80% of leaf nutrients. The withdrawal of chlorophyll during senescence reveals accessory pigments including carotenoids, producing yellow and orange colors, and anthocyanins, producing red and purple colors, creating the colorful autumn displays in temperate forests. The functions of anthocyanins during senescence remain debated, with hypotheses including photoprotection, herbivore deterrence, and pathogen resistance. Evergreen leaves are retained year-round or for multiple years, with gradual turnover rather than synchronous shedding. Most conifers are evergreen, retaining needle-like leaves for 2 to 10 years or more, depending on species and conditions. Many tropical trees are also evergreen, though some are deciduous, shedding leaves during dry seasons. Evergreen leaves allow photosynthesis throughout the year when conditions permit, providing advantages in environments without severe seasonal stress. However, evergreen leaves must withstand multiple seasons of herbivore attack and pathogen infection, requiring greater investment in defenses. Leaf arrangement on stems, phyllotaxis, follows predictable patterns described mathematically. Alternate phyllotaxis has single leaves at each node, positioned at angles relative to previous leaves that maximize light capture by minimizing shading. The most common angle is approximately 137.5 degrees, the golden angle, which is related to the Fibonacci sequence and allows efficient packing. Opposite phyllotaxis has two leaves at each node on opposite sides of stems. Whorled phyllotaxis has three or more leaves at each node, arranged in circles around stems. These patterns arise from developmental processes involving hormone gradients and mechanical forces during leaf initiation. Compound leaves are divided into multiple leaflets, contrasting with simple leaves that have single continuous blades. Compound leaves may be pinnate, leaflets arranged along central axes, or palmate, leaflets radiating from single points. The evolutionary advantages of compound leaves are debated, but may include reducing heat loads through smaller leaflet sizes while maintaining large total leaf areas, facilitating rapid leaf area changes through leaflet movements, and allowing selective shedding of damaged leaflets rather than entire leaves. Many legumes have compound leaves, as do trees including ashes, walnuts, and horse chestnuts. Specialized leaf types evolved for functions beyond photosynthesis. Spines, modified leaves providing defense against herbivores, occur in cacti, barberries, and many other plants. Tendrils, modified leaves allowing climbing, occur in peas, grapes, and other vines. Bulb scales, modified leaves storing nutrients, occur in onions, tulips, and many other geophytes. Pitcher plant leaves trap insects in fluid-filled pitchers, providing nitrogen supplementation in nutrient-poor habitats. Venus flytrap leaves have active traps that close rapidly around prey. These diverse modifications demonstrate the evolutionary lability of leaf form. The internal structure of leaves shows remarkable variation, reflecting different evolutionary strategies. Sun leaves, developing in high light, are typically thicker than shade leaves, with more palisade layers and higher chlorophyll concentrations. Shade leaves are thinner with fewer palisade layers, but larger individual chloroplasts and higher chlorophyll concentrations per chloroplast, maximizing light capture in low light. Some species produce both sun and shade leaves on the same individual through phenotypic plasticity, optimizing leaf structure for local light conditions. Leaf venation patterns, the arrangement of vascular bundles within leaves, vary systematically between monocots and dicots. Monocots typically have parallel venation, with major veins running parallel from leaf bases to tips, while dicots have reticulate or net-like venation, with branching veins creating interconnected networks. These patterns reflect different mechanical requirements and resource transport strategies. Parallel venation provides longitudinal strength suited to elongated grass leaves, while reticulate venation provides more uniform support across broadleaf blades and creates redundant pathways, ensuring continued function if some veins are damaged. The specific leaf area per unit leaf mass, provides a measure of leaf construction strategy. High specific leaf area indicates thin leaves with low mass per area, typical of fast-growing species from resource-rich environments. Low specific leaf area indicates thick leaves with high mass per area, typical of slow-growing species from resource-poor or stressful environments. Specific leaf area correlates with numerous other leaf traits, including photosynthetic rate, nitrogen concentration, and leaf lifespan, creating the leaf economic spectrum where traits vary in predictable ways, reflecting fundamental trade-offs in resource allocation. Trichomes, hair-like appendages on leaf surfaces, serve multiple functions including reducing water loss by increasing boundary layer thickness, reflecting excess light, deterring herbivores through physical barriers or glandular secretions, and absorbing water from fog or dew. The density and type of trichomes vary enormously among species, with some desert plants having dense white trichomes that give leaves a woolly appearance, reflecting light and reducing leaf temperatures. Carnivorous sundews have stalked glandular trichomes that secrete sticky mucilage trapping insects and digestive enzymes that break down prey. Hydothodes, specialized structures that exude liquid water, occur on leaf margins or tips of many species, particularly those growing in high humidity environments. The process of guttation, where liquid water is forced out of hydothodes by root pressure, is most common in the morning when soils are moist and transpiration is low. The exuded water often contains dissolved minerals and organic compounds, appearing as droplets on leaf edges. While guttation resembles dew, it originates from within plants rather than condensing from atmospheric moisture. Vascular tissues transport water, nutrients, and organic compounds throughout plants. The evolution of vascular tissues, xylem conducting water and dissolved minerals upward from roots, and phloem conducting sugars and other organic compounds throughout plants, was crucial for the evolution of large plant size and occupation of terrestrial habitats. Understanding how these tissues function reveals the physical and physiological principles constraining plant performance and structuring plant diversity. Xylem consists of two types of conducting cells: tracheids, which are found in all vascular plants, and vessel elements, which evolved later and occur only in angiosperms and a few other groups. Both cell types are dead at maturity, consisting of hollow tubes with lignified, strengthened, and waterproofed walls. Tracheids are elongated cells with tapered ends and pits, thin areas in walls allowing water movement between adjacent cells. Vessel elements are shorter and wider with perforated end walls, forming continuous tubes called vessels that are more efficient for water transport than tracheids. Water movement through xylem is driven by transpiration, evaporation from leaf surfaces, creating tension that pulls water upward from roots. This cohesion tension mechanism relies on water's cohesive properties, strong hydrogen bonds between water molecules, allowing continuous water columns to be pulled upward without breaking. The maximum height to which water can be pulled is theoretically about 120 to 130 meters at sea level, close to the heights of the tallest trees, suggesting height is limited by water transport physics. However, xylem water columns do break, forming embolisms, gas bubbles that block water flow. Embolisms form when tensions exceed the cohesive strength of water, typically occurring under water stress when high transpiration and low soil water create large tensions. Once formed, embolisms spread to adjacent cells through pits if tensions are sufficient to pull air across pit membranes. Species vary in resistance to embolism, with hydraulic safety quantified as the water potential causing 50% loss of conductivity, ranging from -1 to -2 megapascals in embolism-sensitive species to -10 to -15 megapascals in highly resistant species. Vulnerability to embolism trades off with hydraulic efficiency, the rate of water transport for given pressure gradient. Efficient xylem has wide vessels or tracheids allowing rapid flow, but wide conduits are more vulnerable to embolism because bubbles can expand more easily in larger spaces. Safe xylem has narrow conduits that resist embolism, but narrow conduits reduce flow rates, limiting photosynthesis and growth. This safety-efficiency trade-off helps explain differences in xylem structure among species, with fast-growing species from moist habitats having efficient but vulnerable xylem, while slow-growing species from dry habitats have safe but inefficient xylem. Repair of embolised xylem occurs through several mechanisms. New xylem produced by cambium replaces old embolised xylem, though this requires time and resources. Some species can refill embolised conduits by generating positive root pressure at night that forces water into embolised vessels, redissolving bubbles. Other species isolate embolised conduits by blocking pits, preventing embolism spread while sacrificing conductivity of blocked conduits. The balance between embolism formation and repair determines whether plants experience progressive hydraulic failure during droughts. Phloem consists of sieve tube elements and companion cells that remain alive at maturity but are highly modified. Sieve tube elements have perforated end walls called sieve plates, allowing fluid flow between adjacent cells, forming continuous sieve tubes. The cytoplasm is reduced, lacking nuclei and most organelles, but retains plasma membrane and some cellular machinery. Companion cells retain full complements of organelles and are connected to sieve tube elements by plasmodesmata, providing metabolic support. Sugars are loaded into phloem at source tissues, primarily mature leaves, and unloaded at sink tissues, growing organs, storage organs, and roots, through active transport requiring energy. The accumulation of sugars in phloem creates high osmotic pressures that draw in water, generating positive pressures of 1 to 2 megapascals. This pressure drives bulk flow from high-pressure sources to low-pressure sinks, a mechanism called pressure flow that doesn't require energy for transport itself, only for loading and unloading. Phloem transport is bidirectional and flexible, with flow direction determined by source and sink locations that change seasonally. During the growing season, sugars flow from leaves to growing shoots, roots, and fruits. During spring, stored carbohydrates flow from roots and woody stems to expanding buds and developing leaves. This flexibility allows plants to allocate resources where needed, responding to damage, environmental conditions, and developmental programs. The velocity of phloem transport is surprisingly fast, typically 30 to 150 centimeters per hour, allowing sugars to move from leaves to roots in hours to days rather than weeks. This rapid transport is crucial for coordinating growth of distant organs and for rapidly responding to damage or stress. Phloem also transports amino acids, hormones, signaling molecules, and RNA, allowing communication between organs and potentially coordinating development and responses to environment. Vascular tissue arrangement varies among plant groups and organs. In stems, xylem and phloem are typically organized into vascular bundles, with xylem toward the center and phloem toward the outside. In dicot and gymnosperm stems, bundles are arranged in a ring, with cambium between xylem and phloem allowing secondary growth, increased stem diameter. In monocot stems, bundles are scattered throughout the stem, and there is typically no cambium and no secondary growth, explaining why palms and other monocots don't produce wood. Secondary growth produces wood, secondary xylem and bark, secondary phloem plus periderm. Cambium is a lateral meristem, a cylinder of dividing cells that produces xylem cells toward the inside and phloem cells toward the outside. The rate of cell production is much higher on the xylem side, so wood accumulates while phloem remains thin. In temperate regions, cambium is active during the growing season and dormant during winter, producing annual growth rings visible in cross-sections. Ring width reflects growing conditions, with wide rings indicating favorable years and narrow rings indicating unfavorable years. The formation of heartwood in older xylem represents another important process. As trees age, the oldest xylem at the stem center stops conducting water, and the cells become filled with resins, tannins, or other compounds that resist decay. This heartwood provides mechanical support, but not water transport, which occurs only in the outer sapwood. The proportion of heartwood to sapwood varies among species, with some species having small sapwood bands and extensive heartwood, while others have wide sapwood and minimal heartwood. Heartwood is typically darker and more decay-resistant than sapwood, making it valuable for lumber. The annual growth rings in temperate trees provide remarkably detailed records of past growing conditions, allowing reconstruction of climate, fire history, and disturbance events spanning centuries or even millennia. Dendrochronology, the science of dating and analyzing tree rings, matches ring width patterns among trees to build chronologies extending beyond the lifespans of individual trees. The longest continuous chronologies, based on overlapping sequences from living trees and preserved wood, extend over 10,000 years, providing high-resolution climate records far predating instrumental measurements. Ring anatomy provides additional information beyond simple width measurements. Early wood formed in spring has large, thin-walled cells optimizing water transport capacity to support rapid leaf expansion and photosynthesis. Late wood formed in summer has smaller, thick-walled cells providing structural support. The transition between early and late wood creates visible ring boundaries. The relative proportions of early and late wood, along with variations in cell sizes and wall thicknesses, reflect growing conditions including temperature, precipitation, and day length. Reaction wood, specialized wood formed in response to mechanical stress or displacement from vertical, allows trees to maintain upright growth or bend toward light. Compression wood in conifers forms on the lower sides of leaning stems or branches, with cells having rounded cross-sections and thicker walls containing more lignin, creating compression forces that push stems upward. Tension wood in angiosperms forms on upper sides of leaning stems, with fibers containing gelatinous layers that shrink upon drying, creating tension forces that pull stems upward. The formation of reaction wood throughout tree lifetimes allows continuous adjustment of growth form. Flowers facilitate sexual reproduction through diverse pollination and fertilization mechanisms. Flowers, the reproductive structures unique to angiosperms, show extraordinary diversity in size, shape, color, and fragrance, reflecting adaptations to diverse pollination mechanisms. Understanding flower structure and function reveals the intimate relationships between plants and pollinators that have driven much angiosperm diversification. The basic flower structure consists of 4 whorls of modified leaves, sepals, typically green structures protecting developing buds, petals, typically colorful structures attracting pollinators, stamens, male structures producing pollen, and carpels, female structures containing ovules. This basic plan is highly modified among species, with various organs absent, fused, or multiplied. Some flowers lack petals, relying on wind or water for pollination. Others have petals fused into tubes, restricting access to specialized pollinators. Still others have numerous stamens or carpels, increasing reproductive output. Stamens consist of filaments, stalks, supporting anthers' pollen-producing structures. Inside anthers, microsporangia produce microspores through meiosis. Each microspore develops into a pollen grain containing a generative cell that will divide to produce two sperm cells and a tube cell that will form the pollen tube. Mature anthers dehisce, split open, releasing pollen for pollination. The timing of dehiscence and mechanisms of pollen release vary among species, influencing pollination success and contributing to reproductive isolation between species. Carpels consist of stigmas, receptive surfaces for pollen, styles, stalks connecting stigmas to ovaries, and ovaries, chambers containing ovules that will become seeds after fertilization. Inside ovules, megasporangia produce megaspores through meiosis. One megaspore develops into an embryo sac containing 7 cells, including 1 egg cell and 2 polar nuclei. This highly reduced female gametophyte represents the remnant of the separate free-living generation in non-flowering plants. Pollination, the transfer of pollen from anthers to stigmas, occurs through multiple mechanisms with different ecological and evolutionary implications. Wind pollination, characteristic of grasses, many trees including oaks and pines, and some herbaceous plants, relies on producing enormous quantities of pollen and exposing reproductive structures to wind. Wind-pollinated flowers are typically small and inconspicuous, lacking showy petals and fragrance that would attract animals. Stamens and stigmas are exposed, with feathery stigmas providing large surface areas for capturing airborne pollen. Animal pollination involves diverse animals including insects, birds, bats, and occasionally mammals like monkeys or marsupials. Different pollinator groups favor different floral traits, creating pollination syndromes, correlated sets of traits associated with particular pollinators. Bee-pollinated flowers are often blue or yellow, colors visible to bees, with landing platforms and nectar guides, patterns visible under ultraviolet light that guide bees to nectar. Bird-pollinated flowers are often red, a color visible to birds but not most insects, with tubular shapes matching bird beaks and abundant dilute nectar. Bat-pollinated flowers open at night, are often white or pale, and produce strong musky scents attractive to bats. The most specialized pollination relationships involve obligate mutualisms, where plants depend entirely on specific pollinators and vice versa. Fig-fig wasp mutualisms exemplify this, with over 800 fig species each pollinated by one to a few wasp species. Female wasps enter figs, enclosed in inflorescences, through small openings, often losing wings and antennae in the process. Inside, they pollinate flowers while laying eggs in some flowers, which develop into galls where wasp larvae mature. The specificity maintains reproductive isolation among fig species and creates co-evolutionary dynamics where changes in either partner affect the other. Orchids show extraordinary floral diversity and pollination mechanisms, with approximately 28,000 species employing diverse strategies including food rewards, nectar, pollen, or oils. Food deception, where flowers resemble rewarding flowers but provide no reward, and sexual deception, where flowers mimic female insects, inducing male insects to attempt copulation and incidentally transferring pollen. Some orchids have species-specific pollinators and elaborate mechanisms, ensuring pollen placement on specific body parts, maximizing transfer efficiency and reducing loss to heterospecific stigmas. After pollen lands on compatible stigmas, pollen tubes germinate and grow through stigmas, and styles toward ovules, guided by chemical signals. The tube cell nucleus directs growth while the generative cell divides, producing two sperm cells that travel through pollen tubes. Pollen tube growth can take minutes to days depending on style length and species, requiring substantial resources that styles obtain from surrounding tissues. In some species, pollen tubes compete to reach ovules first, creating selection for rapid growth and potentially allowing mate choice by female tissues. When pollen tubes reach embryo sacs, they release sperm cells in a process called double fertilization, unique to angiosperms. One sperm fuses with the egg, forming a diploid zygote that develops into the embryo. The other sperm fuses with the two polar nuclei, forming a triploid endosperm that provides nutrients for embryo development. This double fertilization ensures that endosperm, which requires substantial resources, is only produced when fertilization succeeds, avoiding waste of resources on unfertilized ovules. Following fertilization, ovules develop into seeds containing embryos, endosperm providing nutrition, and seed coats providing protection. Ovaries develop into fruits, structures protecting and dispersing seeds. Fruit diversity rivals floral diversity, reflecting adaptations to different dispersal mechanisms. Fleshy fruits including berries, drupes, fruits with stony pits, and pomes, fruits with papery cores, attract animals that consume fruits and disperse seeds through feces. Dry fruits including capsules, follicles, and achenes use various mechanisms including wind, water, explosive dehiscence, or animal fur for dispersal. Some plants reproduce asexually through vegetative propagation, producing new individuals from shoots, roots, or leaves without fertilization. Strawberries produce stolons, horizontal stems that root at nodes, generating daughter plants. Many grasses and some trees produce rhizomes, underground stems that produce new shoots. Aspen clones, genetically identical trees connected by root systems, can include thousands of individuals covering many hectares. The largest known organism by area is an aspen genet in Utah called Pando, estimated to include approximately 47,000 ramets covering 43 hectares and weighing approximately 6,000 tons. Vegetative reproduction allows rapid colonization and persistence in stable environments, but reduces genetic diversity and adaptive potential. Self-incompatibility systems prevent self-fertilization in many flowering plant species, promoting outcrossing and genetic diversity. These systems recognize self-pollen through molecular markers and reject it before fertilization can occur. Gametophytic self-incompatibility, found in families including Solanaceae and Rosaceae, involves recognition between pollen and pistil based on shared S-locus alleles, with pollen tubes from incompatible crosses arrested in styles. Sporophytic self-incompatibility, found in families including Brassicaceae, involves recognition at stigma surfaces, with incompatible pollen failing to germinate or penetrate stigmas. Some species show cryptic self-incompatibility, where self-pollen can complete fertilization, but is outcompeted by outcross pollen when both are present. Floral scent involves complex mixtures of volatile organic compounds that attract pollinators, while sometimes deterring herbivores or pathogens. Different pollinator groups respond to different scent profiles. Bees favor sweet floral scents, flies favor putrid odors resembling carrion or dung, beetles favor fruity or spicy scents, and moths favor strong, sweet scents released at night. The biochemical pathways producing floral scents have been elucidated for many compounds, involving modifications of amino acids, fatty acids, and terpenoid precursors. The timing of scent emission often matches pollinator activity patterns, with day-flying pollinators served by diurnal scent emission and nocturnal pollinators by nighttime emission. Nectar composition varies among species and pollinator types, containing primarily sugars, but also amino acids, proteins, lipids, vitamins, and secondary metabolites. The sugar composition, proportions of sucrose, glucose, and fructose, varies with pollinator preferences, with hummingbirds preferring sucrose-rich nectar, while many insects prefer hexose-rich nectar. Nectar concentration varies from about 15% sugar in bird-pollinated flowers to 70% in some bee-pollinated flowers, balancing energy content against viscosity that affects consumption rates. Some nectar contains alkaloids or other defensive compounds that deter nectar robbers, animals that consume nectar without pollinating, while not deterring legitimate pollinators that have physiological tolerance. Plants exhibit diverse movements and behaviors despite lacking nervous systems. Plants, though lacking nervous systems and muscles, exhibit numerous movements and behaviors in response to environmental stimuli. These responses, occurring over timescales from fractions of seconds to years, demonstrate sophisticated sensory capabilities and integration of information that challenge the traditional view of plants as passive organisms. Phototropism, growth toward light, involves differential growth on shaded versus illuminated sides of stems, bending shoots toward light sources. This response is mediated by auxin, a plant hormone that promotes cell elongation. Auxin is produced in shoot tips and moves downward, but light causes auxin redistribution to shaded sides, where higher auxin concentrations promote more elongation, bending stems toward light. This elegant mechanism allows plants to optimize light capture without requiring complex sensory or motor systems. Gravitropism, growth responses to gravity, causes roots to grow downward and shoots to grow upward, even in complete darkness. This response involves statoliths, dense starch-filled amyloplasts that settle to the bottom of cells under gravity. The position of statoliths is detected by as yet incompletely understood mechanisms, triggering auxin redistribution that causes differential growth. In roots, higher auxin on lower sides inhibits elongation, causing downward bending. In shoots, higher auxin on lower sides promotes elongation, causing upward bending. Thigmotropism, growth responses to touch, allows climbing plants to wrap around supports. Tendrils and twining stems respond to contact by growing more on the untouched side, causing coiling around contacted objects. This response can be remarkably rapid, with some tendrils completing one coil around supports in minutes to hours. The mechanism involves touch-sensing proteins in cell membranes that trigger calcium influxes, which activate auxin redistribution and differential growth. Nastic movements are non-directional responses to environmental stimuli, including nyctinasty, daily leaf or flower movements in response to light-dark cycles, thigmonasty, touch responses, and seismonasty, responses to disturbance. Many legumes exhibit nyctinasty, with leaves folding upward at night and opening during day, movements controlled by reversible changes in turgor pressure in pulvini, swollen regions at leaf bases. The adaptive function is debated, but may include reducing nighttime heat loss, reducing herbivore attack, or allowing dew to roll off leaves. Mimosa pudica, the sensitive plant, exhibits rapid thigmonastic responses, with leaves folding within seconds of touch. This movement involves action potentials, electrical signals similar to animal nerve impulses, propagating along leaves and triggering rapid turgor pressure loss in pulvini, collapsing leaves. The mechanism is remarkable in that plants accomplish rapid communication and movement without nervous systems or muscles. The adaptive function likely involves startling herbivores or making leaves less palatable by reducing surface area. The Venus flytrap exhibits even more dramatic rapid movements, with trap lobes closing in fractions of a second when trigger hairs are stimulated. This movement is among the fastest in the plant kingdom, with lobes closing at speeds exceeding 100 millimeters per second. The mechanism involves elastic instability. The lobes are held in metastable states under tension, and stimulation triggers rapid transition to stable closed states, analogous to metal snap fasteners. The trap only closes if two trigger hairs are stimulated within 30 seconds or one hair is stimulated twice, providing filtering that prevents responses to false alarms like raindrops. Circadian rhythms, approximately 24-hour cycles, occur in plants as in animals, regulating numerous processes including leaf movements, stomatal opening, flower opening, fragrance emission, and gene expression. Plants can anticipate daily environmental changes including dawn, dusk, and temperature cycles, adjusting physiology in advance to optimize performance. The mechanisms involve transcriptional translational feedback loops, where clock genes regulate their own expression, with delays creating oscillations. Clock function requires multiple interacting components, with approximately 20 genes identified as clock components in Arabidopsis. The circadian clock confers fitness advantages by allowing temporal coordination of activities and anticipation of predictable changes. Plants with functional clocks show higher photosynthesis, growth, and competitive ability than plants with disrupted clocks or plants in non-24-hour cycles. The clock also gates, restricts to appropriate times, responses to other environmental signals, preventing inappropriate responses to transient stimuli. Plants communicate chemically both within individuals and between individuals. When herbivores damage leaves, plants produce volatile organic compounds, airborne chemicals, that attract predators and parasitoids of the herbivores, providing indirect defense. The same volatiles can warn undamaged parts of the same plant and neighboring plants of impending attack, triggering preemptive defense responses. Evidence for this communication includes increased resistance in undamaged leaves of attacked plants and in unattacked neighboring plants, suggesting information transfer through volatiles. Whether this represents true communication, information transfer benefiting both sender and receiver, or eavesdropping, receivers exploiting information without benefiting senders, is debated. Emitting volatiles benefits attacked plants through indirect defense, but may cost resources and attract additional herbivores. Neighboring plants that detect volatiles and induce defenses clearly benefit. If neighboring plants are genetically identical or closely related, kin selection could favor communication, but volatiles also inform unrelated competitors. The evolutionary stability of volatile emission remains an active research question. Root signaling involves exudation of chemicals that affect neighboring plants and soil organisms. Allelopathy, release of chemicals inhibiting other plants, has been documented in many species, with effects ranging from reduced germination to reduced growth to death. Black walnut trees produce juglone, which inhibits many species, creating zones beneath canopies with sparse understories. The adaptive value is presumably reduced competition, though empirical evidence for fitness benefits is limited. Allelopathy is difficult to demonstrate conclusively because chemicals may have other functions or may be produced during decomposition rather than actively exuded. Root exudates may also convey information about identity, potentially allowing kin recognition. Studies report that some species grow fewer roots or allocate more growth above ground when neighbors are siblings versus non-siblings, suggesting recognition and reduced competition among relatives. The mechanisms could involve chemical detection of exudate differences among genotypes. However, these findings are controversial, with debates about mechanisms, generality, and evolutionary plausibility of kin recognition in plants. Heliotropism, the movement of plant parts to track the sun's position across the sky, occurs in many species including sunflowers, which famously orient their flower heads eastward in the morning and follow the sun westward throughout the day. This solar tracking maximizes light interception and, in sunflowers, increases flower temperature in the morning, which attracts pollinators. The mechanism involves differential growth rates on opposite sides of stems controlled by circadian clock genes interacting with light signals. Interestingly, mature sunflower heads cease tracking and face permanently eastward, providing consistent orientation that optimizes morning warming. Seismonastic movements, rapid responses to mechanical disturbance beyond touch, occur in various plants. The telegraph plant or dancing plant, Codariocalyx modarius, exhibits spontaneous leaflet movements even without external stimulation, with small lateral leaflets rotating in elliptical patterns continuously. The adaptive function of these movements remains unclear, though hypotheses include thermoregulation, deterring insect herbivores, or facilitating rain shedding. The movements are among the most active in plants, occurring continuously throughout the day and responding to various environmental factors, including light, temperature, and humidity. Chemical defenses protect plants from herbivores, pathogens, and competitors. Plants, being immobile and unable to flee from enemies, have evolved sophisticated chemical defenses that deter herbivores, inhibit pathogens, and sometimes affect neighboring plants. The diversity of plant secondary metabolites, chemicals not directly required for primary metabolism, is staggering, with estimates suggesting over 100,000 compounds identified and potentially millions existing across the plant kingdom. Alkaloids are nitrogen-containing compounds, including many famous plant toxins and drugs. Nicotine produced by tobacco and related plants is highly toxic to insects and vertebrates, acting on nervous systems by binding to acetylcholine receptors. Plants produce nicotine in roots and transport it to shoots, accumulating in leaves where herbivory risk is high. Caffeine, produced by coffee, tea, cacao, and some other plants, deters herbivores and may inhibit competing plants through allelopathy. At high doses, caffeine is toxic to most organisms, but at lower doses it affects animal behavior, potentially manipulating pollinators. Morphine and related opioids from poppies bind to opioid receptors in animal nervous systems, producing analgesia and euphoria that have made opium a valued medicine and drug throughout human history. From the plant's perspective, opioids deter herbivores and have antimicrobial properties. Cocaine from coca plants affects dopamine reuptake, producing stimulant effects that deter herbivores. These and countless other alkaloids demonstrate that plants produce chemicals affecting fundamental animal physiology, presumably because such chemicals provide effective deterrents. Terpenoids are the largest class of secondary metabolites, with over 40,000 known compounds including essential oils, resins, and pigments. Many terpenoids are volatile, contributing to plant scents including pine scent from terpenes in conifer resins, mint scent from menthol and related monoterpenes, and citrus scent from limonene. These volatiles deter some herbivores while attracting pollinators or enemies of herbivores. Pyrethrins from chrysanthemums are potent insecticides used in organic agriculture and household products. Azadirachtin from neem trees disrupts insect development and feeding, providing another organic pesticide. Phenolic compounds include diverse structures from simple phenolic acids to complex tannins and flavonoids. Tannins bind proteins, reducing digestibility and deterring herbivores, particularly mammals, whose digestion depends on microbial breakdown of plant material. Tannin-rich plants, including oaks and tea, require specialized adaptations for herbivores to consume them, with some herbivores producing tannin-binding proteins in saliva that neutralize tannins' effects. Lignans, complex phenolic polymers, strengthen cell walls and resist degradation, protecting against both herbivores and pathogens. Glucosinolates from brassicas, mustards, broccoli, cabbage, break down when tissue is damaged, releasing toxic isothiocyanates that deter most herbivores. The two-component defense, where inactive precursors and activating enzymes are stored separately and mix when cells are disrupted, is common, allowing rapid production of toxins without requiring storage of toxic compounds. Some specialists, including cabbage butterflies and flea beetles, evolved enzymes that detoxify glucosinolates, allowing them to exploit brassicas despite defenses. These specialists often use glucosinolates as host recognition cues, demonstrating evolutionary arms races where defenses become enemy attractants. Cyanogenic glycosides occur in many plants, including cassava, lima beans, cherries, and bamboo. When tissue is damaged, these compounds release hydrogen cyanide, a potent toxin inhibiting cellular respiration. Cassava requires processing to remove cyanogenic glycosides before consumption, with traditional fermentation methods detoxifying tubers. The presence of cyanogenic glycosides in foods demonstrates that plants frequently contain compounds dangerous to herbivores, including humans, requiring cultural knowledge for safe consumption. Latex and resins are sticky defensive fluids stored under pressure in specialized ducts. When plants are damaged, these fluids exude and gum up herbivore mouthparts, deterring feeding. Latex from rubber trees is commercially valuable for rubber production, representing a plant defense co-opted by humans. Resins from conifers have antimicrobial properties and seal wounds, preventing pathogen entry. Some insects, including bark beetles, introduce fungi that overcome resin defenses, demonstrating complex tritrophic interactions among plants, herbivores, and pathogens. Induced defenses, defenses produced or activated after attack, represent cost-saving strategies that avoid maintaining expensive defenses when unnecessary. Many plants increase production of defensive chemicals within hours to days of attack, concentrating defenses where and when needed. The signals triggering induction include jasmonic acid, a hormone produced at wound sites, and cystamin, a peptide signal in some plants. Induced defenses can persist for days to weeks, providing short-term protection while allowing reduced investment when herbivory is low. Indirect defenses involve attracting predators and parasitoids of herbivores rather than directly harming herbivores. Many plants release volatiles when attacked that attract natural enemies of the attackers. For example, caterpillar-damaged plants emit volatiles attracting parasitic wasps that lay eggs in caterpillars, killing them. Some plants provide housing, domatia, or food, extrafloral nectaries producing nectar outside flowers, for ants that defend plants against herbivores and sometimes against competing plants. These mutualisms can be obligate, with certain acacias depending entirely on specific ant species for defense. Physical defenses complement chemical defenses. Thorns, spines, and prickles deter large herbivores. Trichomes, hair-like structures on leaf surfaces, can be dense enough to prevent small insects from reaching the epidermis, or can be glandular, secreting sticky or toxic substances. Silica deposits in grasses dull herbivore teeth, reducing feeding efficiency. Tough, fibrous tissues resist chewing and reduce digestibility. Rhaphids, needle-shaped calcium oxalate crystals, stab herbivores' mouths, causing pain and deterring feeding. Tolerance represents an alternative strategy to resistance, where plants maintain fitness despite herbivory through compensatory growth rather than preventing herbivory. Grasses exemplify tolerance, with growth meristems located at stem bases rather than tips, allowing regrowth after grazing removes above-ground tissue. Some plants overcompensate, actually increasing fitness after moderate herbivory, possibly because herbivory removes apical dominance, promoting lateral branching, or because regrowth tissue has higher photosynthetic rates. However, overcompensation is controversial, with debates about how common it is and whether it represents adaptation to herbivory or byproduct of growth patterns. The diversity of defensive chemicals reflects co-evolutionary arms races between plants and herbivores spanning millions of years. As plants evolved new defenses, herbivores evolved counteradaptations, including detoxification enzymes, sequestration of plant toxins for their own defense, and behavioral avoidance. Some herbivores became specialists, adapting to specific plant defenses and using defensive compounds as host recognition cues, creating feedback loops where defenses both protect plants and attract adapted specialists. This co-evolutionary process has driven much of the diversification in both plants and herbivorous insects, with approximately half of all described species being either plants or herbivorous insects. The costs of defense, resources allocated to defensive compounds and structures, create trade-offs with growth and reproduction. Plants in low herbivory environments often evolve reduced defenses, reallocating resources to growth. Introduced plants sometimes escape specialist herbivores from their native ranges, allowing reduction in defenses and increased growth or reproduction in introduced ranges. The evolution of increased competitive ability, or EICA hypothesis. However, defenses may have multiple functions beyond herbivore deterrence, including pathogen resistance, UV protection, or allelopathy, complicating predictions about defense evolution. Plants defend against pathogens through immune responses resembling animal immunity. Plants face constant attack from viruses, bacteria, fungi, and other pathogens, yet most plants remain healthy most of the time, indicating effective immune systems. Plant immunity shares fundamental features with animal immunity, including recognition of enemies, signaling cascades activating defenses, and immunological memory, though implemented through different molecular mechanisms, reflecting independent evolution of complex immune systems. The first line of defense involves physical barriers including cuticles, cell walls, and bark that prevent pathogen entry. Stomata provide entry points for some pathogens, with guard cells closing in response to bacterial signals, providing active defense. Cuticle thickness varies among species and with growing conditions, with thicker cuticles providing better barrier defense but potentially reducing gas exchange. Cell walls contain antimicrobial compounds and can be rapidly reinforced at infection sites through callose deposition, adding polysaccharide barriers and lignification, adding phenolic polymers. If pathogens breach physical barriers, plants recognize molecular patterns characteristic of pathogens or damage. Pattern recognition receptors on cell surfaces detect PAMPs, pathogen-associated molecular patterns including bacterial flagellin, fungal chitin, and viral coat proteins, or DAMPs, damage-associated molecular patterns released from damaged plant cells. Recognition triggers signaling cascades involving calcium influxes, reactive oxygen species production, and MAP kinase pathways that activate defense gene expression. This innate immune response, pattern-triggered immunity, provides broad-spectrum resistance against many pathogens, though some pathogens evolved effectors, molecules that suppress pattern-triggered immunity, allowing infection. In response, plants evolved resistance genes encoding receptors that recognize specific pathogen effectors, triggering effector-triggered immunity, stronger responses including hypersensitive response, rapid cell death at infection sites that deprives biotrophic pathogens of living tissue they require. This creates evolutionary arms races where pathogens evolve new effectors to avoid recognition, and plants evolve new resistance genes recognizing new effectors. The hypersensitive response involves programmed cell death at infection sites, visible as necrotic lesions surrounded by healthy tissue. This sacrifices infected cells to prevent pathogen spread, analogous to amputating infected limbs. The cell death is active and controlled, involving specific signaling pathways and gene expression, not passive tissue death. Reactive oxygen species and antimicrobial compounds accumulate at infection sites, directly killing pathogens and signaling defenses in surrounding cells. Systemic acquired resistance provides immunological memory, with local infections inducing resistance in distant uninfected tissues that persists for weeks to months. The signal molecule is salicylic acid, related to aspirin's active ingredient, which accumulates at infection sites and moves systemically, priming distant tissues for rapid defense responses. Primed tissues show faster and stronger responses to subsequent infections, providing adaptive immunity-like protection. This demonstrates that plants can remember past infections despite lacking adaptive immune systems like those in vertebrates. RNA interference provides antiviral defense, with plants producing small RNAs complementary to viral RNA that guide degradation of viral genetic material. This mechanism is highly specific, only affecting targeted viral sequences, and can provide systemic protection through movement of small RNAs through phloem. The discovery that plants use RNA interference for antiviral defense inspired biotechnology applications, including engineered virus resistance, by expressing hairpin RNAs targeting viral genes. Pathogens cause tremendous damage to crops and natural vegetation, with plant diseases reducing crop yields by an estimated 20 to 40% globally despite extensive pesticide use. Some diseases have caused historical impacts, including potato late blight, causing the Irish potato famine in the 1840s, killing approximately 1 million people and causing mass emigration. Chestnut blight essentially eliminated American chestnut as a canopy dominant in eastern North American forests in the early 20th century. Dutch elm disease killed millions of elm trees in North America and Europe. These examples demonstrate the vulnerability of plants to introduced pathogens against which they lack co-evolved resistance. Managing plant diseases involves multiple strategies, including resistant varieties, breeding or genetic engineering for resistance genes, cultural practices, crop rotation, sanitation, planting density, biological control, using antagonistic microbes or hyperparasites, and chemical control, fungicides, bactericides. Integrated disease management combines these approaches, aiming for sustainable long-term control rather than relying solely on chemicals. Understanding plant immunity at molecular and ecological levels improves ability to develop resistant crops and reduce pesticide dependence. Endophytes, microorganisms living inside plant tissues without causing disease, represent complex relationships ranging from mutualistic to commensalistic to latent pathogenic. Many endophytic fungi and bacteria provide benefits including enhanced stress tolerance, pathogen resistance through competitive exclusion or antibiotic production, and growth promotion through hormone production or nutrient mobilization. Some endophytes produce bioactive compounds with pharmaceutical potential, including the anti-cancer drug Taxol, originally isolated from Pacific yew, but later discovered to be produced by an endophytic fungus. The diversity and functions of plant endophytic communities remain areas of active research. Mycorrhizal associations, discussed earlier in the context of nutrient uptake, also provide disease resistance by occupying root surfaces and excluding pathogens, producing antibiotics and priming plant immune responses. Plants associated with mycorrhizae often show enhanced resistance to root pathogens compared to non-mycorrhizal plants, with resistance varying depending on fungal species, plant species, and pathogen types. Some mycorrhizal fungi produce compounds directly toxic to pathogens, while others stimulate plant defense responses through signaling molecules. The integration of mycorrhizal benefits for both nutrition and disease resistance makes these symbioses crucial for plant health in natural and agricultural ecosystems. If you're still awake somewhere in this exploration of plants, of their evolution and photosynthesis, of their roots reaching deep and leaves reaching high, of their flowers and their defenses, of their movements and their immune systems, there's more to discover, more quiet facts about these ancient organisms that preceded us and that make our lives possible. But if sleep has found you, if consciousness has faded somewhere among the green, then rest knowing the plants continue their patient work. While you sleep tonight, plants continue photosynthesizing wherever sunlight reaches leaves, converting light into chemical bonds that support not only plant growth, but the entire web of life. In darkness, they continue respiring, breaking down sugars to fuel maintenance and growth, preparing for dawn when photosynthesis will resume. Their roots continue growing through soil, exploring for water and nutrients, extending the vast hidden networks that match or exceed above-ground biomass. The plants growing outside your window have no awareness, no consciousness as we understand it, yet they sense their environments with sophistication rivaling any organism. They detect light direction, wavelength, and intensity, adjusting growth and development accordingly. They sense gravity, growing roots down and shoots up without nervous systems or sensory organs. They detect touch, winding around supports or recoiling from disturbance. They monitor temperature, tracking seasons and anticipating frost. They measure day length, timing flowering and dormancy to appropriate seasons. This sensory capacity without awareness, this complex behavior without intention, reveals that much of what organisms do requires neither consciousness nor nervous systems. Plants demonstrate that sophisticated responses to environment can emerge from cellular and biochemical processes distributed throughout organisms rather than centralized in brains. They remind us that life takes many forms, with different solutions to the challenges of survival and reproduction. The evolutionary history of plants spans over 450 million years of adaptation and diversification. From the first small pioneering plants colonizing barren rock to the complex ecosystems of modern forests, grasslands, and wetlands. Every major group of land plants that evolved still persists today: mosses and liverworts in moist habitats, ferns in temperate and tropical forests, conifers in boreal and montane zones, flowering plants everywhere. This persistence suggests each group represents successful solutions to different environmental challenges, with no group completely superior across all conditions. The chemicals plants produce, secondary metabolites evolved for defense and communication, have profoundly influenced human history and culture. Caffeine, nicotine, morphine, cocaine, and countless other plant compounds affect human physiology and behavior, becoming valued medicines and dangerous drugs. Spices derived from plant secondary compounds drove exploration and trade, with the search for pepper, cinnamon, cloves, and nutmeg motivating voyages that connected continents. Understanding plant chemistry provides both practical knowledge for utilizing plants and deeper understanding of the evolutionary processes that generated this chemical diversity. The relationship between plants and the rest of life is fundamental and pervasive. Plants produce the oxygen we breathe, fix the carbon that forms our bodies, create the habitats where we live. Without plants, terrestrial life as we know it could not exist. The landscapes would be barren rock and sand, the atmosphere would lack oxygen, the climate would be harsh and extreme. Yet plants need other organisms too: pollinators to fertilize flowers, dispersers to spread seeds, mycorrhizal fungi to supply nutrients, and even herbivores whose feeding sometimes stimulates compensatory growth. The next time you see a plant, consider the complexity hidden beneath the surface. The cells and tissues and organs working together, the photosynthesis occurring in chloroplasts descended from ancient bacteria, the vascular system moving water and sugars throughout the plant, the roots exploring soil in partnership with fungi, the defenses protecting against countless enemies, the hormones coordinating development and responses to environment. Consider that this living organism shares a common ancestor with you, that the same fundamental processes of genetics, metabolism, and evolution that shaped your lineage also shaped plants, though taking different paths leading to radically different organisms. Plants offer a perspective-expanding view of what life can be, sessile rather than mobile, autotrophic rather than heterotrophic, generating their own food from sunlight rather than consuming other organisms. They demonstrate that evolutionary success doesn't require speed, intelligence, or intentionality, but rather effective solutions to environmental challenges and efficient conversion of available resources into offspring. In this, plants have been extraordinarily successful, dominating terrestrial biomass and primary productivity, while remaining largely unnoticed and underappreciated by human consciousness that focuses on movement and intention. The remarkable thing about plants is not just what they do, but how long they've been doing it. Plants have been photosynthesizing, growing, reproducing, and adapting for over 400 million years, surviving multiple mass extinction events that eliminated dominant animal groups. They witnessed the rise and fall of the dinosaurs, the breaking apart of Pangaea, the formation of mountain ranges and oceans. The basic mechanisms of photosynthesis, the fundamental structure of cells and tissues, the core strategies of defense and reproduction, all have been refined over evolutionary timescales that dwarf human civilization. Consider that the oldest living organisms on Earth are plants. Bristlecone pines over 4,800 years old, predating the pyramids. Aspen clones potentially tens of thousands of years old, persisting since the last ice age. And seagrass meadows in the Mediterranean estimated at over 100,000 years old, making them possibly the oldest living organisms known. These ancient individuals have witnessed the entire span of recorded human history and beyond, growing slowly, steadily, patiently while civilizations rose and fell around them. See you next time.